Hello, students. Welcome to the second episode of the grade 10 science syllabus, mm, English medium, and also Sinhala medium. Uh, I'll teach in both English and Sinhala, and you guys can uh, work on like listen and understand. And also, there's a new update that I'm composing a workbook for the students, like grade 10 and 11 students. Hopefully it'll be there by like from few months, a grade 10 workbook, science workbook. So I hope you guys can buy it after it's available because I'm pretty sure it's gonna be very helpful for your ordinary level exam. Because as I said, I did ordinary level exam like few months ago, and I got an A for science. And while I'm also sharing my tips and tricks for how I did for O levels, I think I can help you guys with um, yeah, the syllabus as much as possible. I will help you guys. So today we have about proteins because we finished about carbohydrates last time. Today we have proteins. So we can start from this part onwards. So I am strictly referring the textbook because I think I mentioned this earlier as well, but the textbook is what's most important in the ordinary level exam. Nothing else from the textbook will come. And just even listening to what I'm saying, and if you guys listen to it carefully, I'm pretty sure that you guys can like easily get like B without studying even. So just listen to what I'm saying carefully. So, if you read this, we can see protein is an essential constituent in all living cells. It's the same as carbohydrates. Proteins are very important for our living cells, living body. So, there's a difference. So, in carbohydrate, do you guys remember what we had? We had carbon, hydrogen, and oxygen, right? There were like only three components present in carbohydrates. But when we come to proteins, we have four that's present. Four and also an additional one that is very optional, but it may be there. So carbon, hydrogen, oxygen, these three are always there in proteins. And more than this, nitrogen is also there. So the four compounds in like elements in the um protein compound is carbon, hydrogen, oxygen, and nitrogen. And also, as I said, there's an optional one named sulfur, S, in the periodic table. It may be present, but it's not like it's not like a must. It's not like there is, but there will be, there might be. So you can see 17% of the mature human body is consisting of proteins. So not much like carbohydrates, but proteins are also an important part in our body. And as we go through, we'll see why it's important in our daily lives, right? So this is not a very big part. And you can see, so this um proteins are made of amino acids. So the like for carbohydrates, you remember, right? You learned glucose, fructose, and galactose, and those stuff, right? So glucose was the structural unit of carbohydrates, right? So for proteins, the structural unit is amino acids. So amino, this is not like very like what do you call it, must to learn, but let me take you guys downwards. So, oh yeah, proteins you guys know, right? So it's in meat, fish, eggs, that we eat. And you say like, oh, I eat meat, I eat eggs, because I personally, in my school here in Korea, I like, so in the morning we always have breakfast. I eat breakfast from my school. So when like because I eat a lot of eggs, my friends are like, "Oh my God, Mahini, you're getting a lot of protein." So yeah, because protein is good for muscles, like muscle gain, and it's like a good source of energy because first when you get like carbohydrates, it's the first thing that gives you energy, and the second would be lipids. Proteins are third, as I remember. Somehow carbohydrates is the most important for like strength. But proteins are in meat, fish, eggs, and stuff. So this is like extra knowledge, as I said before as well. You can see this is like in the picture, it's the structural unit, like how it's composed. So you can see these are like named as an amino group and a carboxyl group. So you can see in the R, it has carbon and hydrogen. So there might be differences of R. You can see 20 different amino acids are present due to the change in R group. So there might be changes if when the R group changes. But this group contains of carbon and hydrogen. So you can see all proteins present in organisms from bacteria to human are made of these 20 different amino acids combined in different sequences. So there is a difference in like when you're combining the amino acids and stuff, it's like different. So they make like various patterns. Um, you can see the simplest amino acid is glycine. H is present as the R group. So as you see, this part, this R group can be consisting of H and C. And they say like simplest amino acid is glycine. So the R group consists of hydrogen in this part. So you can see this is the image of glycine. Oh, 1 second. Yep. Some amino acids cannot be synthesized within the body. So they have to be taken from the outside of food. Therefore, they are known as essential amino acids. Guys, these are not mandatory for you to learn, like you to like study. But at the same time, I think like if you can study this stuff, it'll be really helpful for your A levels and even like going further because like biology, chemistry is not like a subject you just learn for like 1 day and you're like, okay, I'm done with the exam. I don't need it anymore, right? So if you plan to learn more, I think just these extra knowledge parts are really like exciting as well. I love these parts. So when you go for this is not, like, don't study this. Proteins present in muscles, what's the knee? Myosin, actin, bones, osane, like that. I think you guys know what proteins present in red blood cells, hemoglobin, and also hairs, it's keratin. Because you probably have heard, oh, like your parents saying, oh, keratin, you need keratin, and even in ads they're like, oh, keratin, protein and stuff, right? So yeah, keratin is also a protein. So like you don't have to remember those stuff. So as I said, proteins play an important part in our day, like our body in a living organism. So the main thing of about proteins is they provide energy. So as I said, remember that I said carbohydrates and lipids provide the first two energies, right? So there's this thing, imagine like, okay, so you don't eat for days. Okay, so like you eat and like you stop eating at one point and you starve for days. So first your carbohydrates are going to be used up and then lipids are going to be used up. And next, because like the body is finding sources to live, right, to get energy from. So as this days goes by, as carbohydrates get over, lipids get over, the next thing that they would extract is protein. So from the protein particles that's stored in your body, they extract you, like extract them and gets energy to live. So it's a very important energy source as well. And also making structural components. So there's a thing like, have you guys heard of cell membrane? It's like in the cell, inside of the cell. It's not the cell wall, because cell wall we remember we discussed before that cell wall is in the plant cell and that it's consisted of carbohydrates, right? You guys remember, right? Let me show you guys. Yeah, cellulose. You remember, right? Cell wall of plant cell in fibers. So, but this thing is not the cell wall that we are talking about in proteins. Let me draw it and show it to you. So in the cell, a cell looks like this, right? And this is a plant cell. So this is the cell membrane, and this is the cell wall. So this is the one that consists of carbohydrate cellulose, and this is protein. Because they might ask you about, because a lot of people get confused in the cell membrane and cell wall part, because even I got confused. But in the exam, they might ask, okay, what is the cell membrane made of? What is the cell wall made of? And more questions when as we learn further. So it's very important for you guys to remember that. And then hairs and feathers are also in keratin. As I mentioned, keratin is a very important part in our hair. It adds you from keratin protein to protect that, to increase that, like increase the growth, to remove damaged hair, like keratin treatments and stuff. So yeah, keratin is one of the like structural components. So it's a basic um protein. And as enzymes, you can see. So enzymes is known as a catalyst. So what does a catalyst mean? That means it eats up without wasting its energy. You could say that. So enzymes, if you take, it's like um cat a catalyst, as I said, it eats up without like losing oneself. So this enzyme does what it does is it but like it reacts with like in an organism, and then it produces um like it catalyzes the enzyme catalyzes biochemical reactions in an organism. So these enzymes are also known as proteins. Then as hormones, you know that in your body there's a lot of hormones, right? That it stimulates like various processes. So here we can see involving homeostasis and coordination of organisms. So hormones are a really important part in our body, and enzymes are pro like sorry, uh hormones are kind of proteins made by CHON. What is CHON? Carbohydrate, carbon, hydrogen, nitrogen, and oxygen. And as antibodies, you guys know what antibodies are, right? So I remember when I was like in seventh or eighth grade, we learned about antibodies. So antibodies are this like stuff that's produced in our body to act against the diseases or microorganisms that might enter our body. So any like object that is like an external object that comes into our body, my antibodies are ready to fight with it. And it's like it's basically in our immune system, so that it creates a protection to our body. So these antibodies are also like part of a protein. So remember that protein is very important as because its antibodies are also proteins. So you can use a test to identify proteins. I hope you guys remember what we did for carbohydrates. So Oh, I think I didn't do the test to identify carbohydrates, right? So let me walk you through it. So for test to identify carbohydrates, so this differentiate from compound to compound. So for carbohydrates, there are three tests: the starch test, the glucose test, and the sucrose test. So this is a monosaccharide. This is a disaccharide, and this is a polysaccharide, right? You guys remember, right? So yeah. So in the starch test, what we are going to use is known as iodine solution. Okay, so an iodine solution is used. So what we are going to do is in this so we are going to take like maybe bread. Think that we are going to take a little piece of bread and we are going to grind it and put water into it. Like So, you should grind it with water, like not like a very like sticky part, like a bit of water, so we can identify it clearly. So then after like grinding it with water like the small part of like small amount of food, then you can add iodine solution into it. So this iodine solution is like brownish color, and then this color turns into purplish blue when you add like iron like when you add like iron solution to the when you add iron solution to starch, it turns purplish blue. Let me show you guys the iodine solution. So this is the iodine solution that you see. And starch test, if you take what the result would be, yeah, you can take this as well. So this is the purplish blue color that you see. So don't think about this. This is the starch, the positive test. So yeah, you can see it here as well. Dark blue, so it's like purplish, right? Purplish blue, because that's what our book mentions. And if you don't write like purplish blue, you might lose points even if you say just blue. So just write purplish blue after iodine solution is added. Then you have the test for glucose. So glucose is like the structure unit of carbohydrates, right? You guys remember that. So what we do is we take glucose into a test tube, and for this we use the Benedict solution. So remember, for the starch test we use iodine solution. For glucose we use the Benedict solution. So after we get a little bit of glucose into a test tube, we add the Benedict solution into this. Then we put this, so we take this, so imagine this is the test tube and this is glucose, right? Okay, so this is glucose and we add Benedict solution, B Benedict solution, I'm gonna write BS. And then we are gonna keep it in a water bath and heat it. So this is what it basically looks like. And after we heat it, there's a series of color differences that you can see. Guys, this is very important because you might be asked to fill blanks and stuff in the exam from this. So remember, at first, because the Benedict solution is blue, it's blue, then green yellow, then orange, and then brick red precipitate. So this is how it will be arranged as the water bath and everything. Then under it is the candle. So let me show you guys how this glucose test is conducted. Glucose test. Sorry, guys. Yep, so you can see. So this is 0%, blue, green, green yellow, orange, and brick red precipitate. It doesn't show the exact color of what we want, but you guys should write what's in the book, this part, because that's why you get marks for it. So yeah, this is like how it is and how it looks like. And the next test is for sucrose. So sucrose is a disaccharide. I hope you guys remember. So what we do is we take this sucrose into another test tube. And this is also tested by Benedict solution. So remember that the monosaccharide and the disaccharide is checked with the Benedict solution and starch with iodine solution. So as usual, what we do is get sucrose, we add the solution, Benedict solution, and then we keep it in a water bath and heat it. So we don't directly heat it, but we heat it like indirectly using a water bath. And first, you'll see that when you use disaccharides, there will be no change, color change. So this is when the difference occur than the monosaccharide part. So we use diluted sulfuric acid, H2SO4. Even if you write diluted H2SO4, that would be enough for your exam. So as added to a freshly prepared sugar solution and heated. So what we do is this is the first step. This is like a trial method, but it's not a must to do this. What we are going to do is after this part, so what we are going to do is we take sucrose, we add a few bits of H2SO4, diluted H2SO4 into that, and we immerse it in a water bath and heat it. And then we are going to add the Benedict solution into the other one again. And that's when you can see the color change. So with the Benedict solution, there's a different thing in here is H2SO4, like the diluted sulfuric acid. This is very important for you guys to remember because you might forget this for sure because I did. And it's the same color change, blue, green, green, yellow, orange, and black red, brick red precipitate. So that's about the carbohydrate tests. Now let's go into the protein test. There's a lot of stuff, guys, because grade 10 first lesson is like the longest lesson, I guess. And I used to hate it because, like there's a lot of things to study, and I hated studying. Because I personally am someone who like does not like to study. I'm like basically like I hate studying. Like honestly, I hate studying. Even right now, I don't like study much. I just listen to what my teacher says and put it into my head. So it makes things easy, but at the same time, I hate studying so much. So what we are gonna do is the next part is the test to identify proteins. So we call this protein test as butyrate test. So remember for starch, what we use was iodine, and for the other two, we use Benedict, right? So this one is called a butyrate test, the protein test. So for this, you have to get a solution like solution made of any type of protein. So here we can take grinding dal or egg white. Oh guys, remember in an egg, the white part is the one that has protein, not the yellow part. I remember teachers were emphasizing this to me when I was in when I was in like 10th grade. I don't know why, but yeah. So this is obtained like either a white part of this thing or this grinding dal part. And you can see we are going to use sodium hydroxide and copper sulfate. So let's see what this, oh, I can show you guys the, yeah, this is like the test. It's called the Benedict's test. And this is the burette test. Yep, so you can see. So in the burette test, what we do is we take the proteins to one, like either dal or egg white into this thing and then like into a bowl. And then we pour in the in sodium NaOH and CuSO4. So this thing and copper sulfate, like to obtain the reaction. And then you can see this turns into purple color, like the egg white or the grinded dal. Then we have enzymes. You guys, excuse me because I couldn't have dinner, so I'm having dinner while I'm doing this. I'm just eating some candies. Sorry about that. So in the enzymes, what we are gonna see is, oh, sorry about that. So now let's move on to enzymes. So we discussed that enzymes are types of proteins, right? So it's a catalyst. So here you can see a catalyst is like it eats up themselves. Like if you read this, you can see special proteins, organic catalysts. So you can see that they are organic, produced within the organisms to increase the rate of biochemical reactions are known as enzymes. So make sure you study this part because they honestly can ask you this part like very, there's a big possibility that you guys can get this question. Second. So make sure to study this, what an enzyme is, the definition, because no matter how you study, like how you know the definitions, you must know the exact definition of the book. That's where they give marks. So make sure to study what an enzyme is, the special protein or the organic catalyst produced within the organism to speed up the rate of reaction of rate of biochemical reactions. Biochemical reactions. So what we mean by this biochemical reactions, imagine that you're making it from sucrose to glucose. So you're gonna break, right? So this catalyzation generally means that you're gonna break something into something. So here we can see, for example, to convert sucrose into glucose, you know that sucrose is one a disaccharide and glucose is a monosaccharide. So a disaccharide to a monosaccharide. So we are going to heat it with a dilute acid to make it to a monosaccharide. But you can see that if we take as an example in our stomach, like we have the digestive system, right? So how do they make this like food that goes inside our body absorb and give us energy, right? There's a question in that. So these catalysts are the main part. So these enzymes, so there's an enzyme present in the digestive system that do the same reaction, but there's a difference that it does it at a low temperature. So enzymes do the same reaction. That means breaking down sucrose into glucose, but at a lower temperature. So the activity of enzyme is to catalyze the biochemical reaction. So biochemical reactions is basically breaking down the food and all. So next we can see second. Mm, there's like activity you can see that you can use to see like the reaction of amylase on starch. So what we are going to do is we are going to take a test tube as usual and put 2 milliliters of starch solution. So after we put this 2 milliliters of starch solution into the test tube, we add 2 milliliters of amylase. So you know that like let's think of it as a solution of ground germinating green gram like seedlings. So the green like germinating green seeds also like consist of these enzymes. So you can use it as well. So we say that amylase is an enzyme that acts up. So you'll see it in a minute. So add like take the starch and then add amylase into it and mix it well. So then after we get like a drop, so after like shaking it like mixing it, we are gonna take a drop of this solution and keep it on a clear white. Why do we use this white tile? Is to observe the reactions clearly. So we put the solution onto the white porcelain tile, and for this as well, we use iodine because we use starch. We are going to use iodine as well to test this enzyme reactivity. So add a drop of iodine into the like mixture that we made starch and germinating seeds like amylase. And continue same procedure for 20 minutes in 2 minute intervals. So like from 2 minute intervals, you have to continue it for like 20 minutes, which is like 10, 9, 10 times. And then you can see the blue color of the drop obtained from the mixture. So from the first time when you obtain it will be blue color. And as time goes by, you can see that the color, like the blue color that you obtain is turning to the color of iodine, which is like yellowish, brownish, like something like that color. So starch gives black blue color with iodine, but it does not give color change with iodine after 20 minutes if there's no starch. So that's an important point to note. So if there is starch, it does give us a reaction that makes like blackish, purplish blue color, blackish blue, you could say. But if there's like no starch remaining, because as time goes by, the starch will get warmed up, right? Because it's getting over. So after that, 20 minutes, like you won't get any reaction because there's no starch left. And we can see as what the reason for this is that starch is not there is because this starch is going to be converted into maltose. And you can see this enzyme amylase is going to be the part catalyzing this reaction. So, as I said, no starch remaining. That means starch has been catalyzed by the catalyzed to form maltose, and this is done by the amylase enzyme. Moving on to lipids, and I think lipids is the last part for today's session. I know it's like kind of slow, and you guys like might be kind of doubtful why is she like stopping, but it's like as you see, it's like 11:25 p.m. here. And because I'm in the dome, I have like restrictions that I can't do this and that after like this time. So I have to stop it here, but I'll make sure that the next day we will be done with the topic because we did a lot today, right? We did the proteins, we are doing the lipids, and we're also talking about enzymes and the reactivity, like reaction, like activities and all. So it's this lesson is actually kind of hard, but I mean, if you guys want, I can do like this part and a bit of the second unit because I'll do the second unit in the next video, I guess, because the second unit is actually very easy because it's physics. It's just one or two stuff that you have to know. But this is biology, so I hope you guys stay with me until the end because it's actually very big. Like it's not hard, but it's very big. You get what I mean, right? So let's go to lipids. Okay, guys. So what do you think when we hear the name lipids, right? Like we use it daily in daily life, right? So we use lipids usually in just think in our kitchen, like oils. We put oils into it, right? Those stuff. So lipids are divided into fats and oils as well in this, but this is not the lipids that we are like talking like the burning oil, but it's the stuff that's inside our body to produce energy and give us strength. So, what this lipid and like lipids divide into fats and oils. What this fats and oils are like fats are when like the lipids are in solid like room temperature and it's solid like in the room temperature it's solid. It's the fats, and when it's liquid it's called the oils. So remember that it's important why like lipids divided into two like fats and oils because in room temperature this is solid and this is liquid. So as usual, there is carbon, hydrogen, and oxygen as elements of this lipid, but you can see that oxygen is less than in carbohydrates. You might not be questioned on this part, but just remember that in oxygen, like in carbohydrates, there's much more oxygen and there's less oxygen in lipids. And lipids are also insoluble in polar solvents, including in water. That means it doesn't dissolve in polar solvents. And as we go by, we'll understand what water, like polar solvents are, and why water is not a polar solvent. But just remember that lipids are insoluble in polar solvents. But they are soluble in organic solvents. So organic is like that contain carbon. And you can see groundnut. So if you guys want, I can show you organic solvents. Mm, yeah, these are like benzene, xylene. These are like polar solvents, organic solvents. So it's basically the idea is containing carbon. So as examples, groundnut, coconut, gingerly, butter, margarine are examples. Uh, if I show you how it's made, fatty acid and three glycerol, sorry, glycerol and three fatty acids. This is the glycerol molecule, and these are fatty acids. Yeah. And these will be broken down into lipids plus H2O. Mhm. So basically what the idea is, this is the fatty acids and glycerols. This is it. It breaks down to lipids and water. So it's like basically making up actually. So it releases a water molecule as well when there's like when the fatty acids when it gets joined like this. So this is the glycerol and fatty acids. When they get joined, this is a lipid particle and it releases an H2O molecule. So next we can see the significance of lipids. So what as we mentioned before, even in, you remember in the protein parts as well, right? So lipids are an energy source which is used after carbohydrates. Um, you can see that there's a lot of energy producing when you burn lipids. Form different like structural components as well, like carbohydrates and proteins. So that is in the cell membrane as well, phospholipids and cholesterol. So cell membrane, I hope you remember, proteins are also in cell membrane, but remember cellulose is in cell wall. For conservation of water, what this means is to like preserve water. That means keep the water into that itself. So the wax known as Q10, there's a wax called Q10. So it's in the like plant body. So this Q10 is what is a like lipid. It controls the water without letting it go outside and preserve it. And also we can see it in like animals' bodies. So it's also impermeable to water. What impermeable? What do you know what impermeable means? Make sure to comment down what impermeable means. So it's like really important in science to know what the meaning of impermeable means. Uh, Make sure to comment down, and at the end of the video, I'll tell you guys what impermeable and semi-permeable is. And then next we have to regulate, maintain body temperature. I feel like we learned this in like ninth, 10th grade, and no, ninth grade, I guess. Regulate. So the warm-blooded animals, as like, so you remember the warm-blooded, right? We learned it in ninth grade. It's like birds, and we are warm-blooded. There's a fat layer called hypodermal fat layer. It's also like an insulated. What a thermal insulator is like, it makes us like, it doesn't make us feel super hot, like super hot or super cold. Like it regulates the body temperature. So as it says, it helps to maintain the body temperature. So it makes like it protects our body from like various, like extreme coldness and stuff. So lipids are very important in this process of maintaining the body temperature as well. Also, in to protect internal body organs and absorb external shocks. So protection in general. Uh, Also synthesize some hormones. So we can see some hormones of vertebrates: estrogen, testosterone, cortisol, estrogen. So estrogen is like, it's in the female body. Testosterone is in the male body. And you'll get to learn these hormones more when you go further. Somehow remember that there are like lipid compounds. Oh yeah, I didn't say about semi-permeable and impermeable. So impermeable is basically, it does not allow water to pass through it. So what we mean by here is avoid desiccation, as it is impermeable to water. So this wax is impermeable, means it does not allow water to pass by. And semi-permeable is, it allows some substances to pass by, but not everything. These two words are very like popular in the biology like parts. So make sure to remember it. And then test to identify lipids. We call it a Sudan 3 test. So we are going to take a little bit of oil lipids to a test tube, and then add the Sudan 3 region into it. And then you can see red fat globules. Let me show you guys. Sudan test. Yeah, it's basically this. This is the Sudan 3 test that you will see. So just remember that when you add Sudan 3 region, it turns red fat globules. Because guys, this tests are really important for you to study. So make sure you study those. Fatty acids. So this is extra knowledge, but just remember it's like really fun to remember this stuff. But somehow, fatty acids are divided into two parts as saturated and unsaturated. So what saturated means there's only like single bonds between the carbons. So you know that in the lipids there are CHO, right? Carbon, hydrogen, and oxygen. So within this carbon-carbon particles, there can be single or double bonds, or like like because if you take elements, they have various bonds, triple bonds, quadruple bonds, and stuff. So this if there's single bonds between carbon atoms, it's called saturated fatty acids. And you can see it's like solid or semi-solids, which means it's basically a fat in room temperature. And unsaturated is basically there are double bonds. So let me draw. So if a single bond looks like this, but a double bond looks like this. Yeah. So yeah, this is that. So if there's like one or several double bonds within carbon, it's called as unsaturated and it exists. So it's an oil. As I said, oil in room temperature is basically unsaturated fatty acids. I think that will be enough for today for you guys as well because it's a long video, and I'll make sure that I do the distance displacement and physics part for you guys next, because I'm pretty sure like some of you guys are like struggling with the physics part, but don't worry guys, because the physics part is really easy in this. So I'll make sure to help you guys out with the physics part in the next video. And I'll make sure to bring out the next part of this video real quick so we can end this chapter as well. So I hope you guys got a lot of stuff from today's video, and make sure that you guys subscribe and like and comment any questions that you have. I mentioned my number and you can text me and ask any questions that you have as well. And yeah, that will be great. And thank you so much for listening. Have a nice day.